Of the questions that were assigned as individual questions, number 2 was the hardest question. It involved at least 4 parts. The first part has to do with this 3' and 5' end. That has to do with the direction of the molecule. Remember that the reading of the sequence is done from the 5' to the 3' end. So it could only be read starting at the 5' end and then moving towards the 3' end. That's important for how the molecule works. In order for DNA to do anything, the mechanism that replicates DNA, the mechanism that makes proteins, has to be able to read the sequence, has to be able to read the information. And remember, we talked about information already. We looked at information as being differences in codes that allow for specific ideas to be expressed and communicated while others weren't. So this idea of direction is important, just as it is important that you have to read a word from left to right or a paragraph from left to right versus from right to left, making the word or the paragraph meaningless. The next important part that you really have to consider is the matching of the DNA strands and the concept that the two strands are opposite each other. So the codes, the sequences that again are different, the differences in the nitrogenous bases, those codes, those sequences are on the opposite strands, are in fact opposite of one another. This is important for the functioning of DNA. So when we're looking at these strands, the word anti-parallel comes into play. Anti-parallel just means that they are, in a sense, parallel geometrically, but they're opposite in their sequence, so they're opposite in one another. So anti means opposite, but they're also parallel in the sense that their one strand is right next to the other strand. So here's the DNA backbone, that whole ribose, phosphate, sugar, phosphate, the DNA backbone that's made of the sugar phosphates all the way up and down the DNA on the right. And here's another DNA molecule on the left, phosphate, sugar, all the way down. Remember that we're going to read the sequence from 5' to 3'. So if we read this strand from 5' to 3', we would see that the sequence would be T, G, C, A. Now that again is on the left side of the double-stranded DNA is the sequence T, G, C, A, because the code is read from 5' to 3'. So the 5' being T, the next one G, the next one C, and the last one A. Now they match up anti-parallel, so the 5' matches up with the 3', the 3' with the 5'. Now on the opposite side, if we reading this side, that looks like it should be opposite, like if this is T, this is A, if this is G, this is C. So this sequence should, if you just read it across, would be different from this sequence. But if we look at it in the sense of direction, again, we're looking for the word. That word direction is so important when you're talking about information. It's not just differences in information and differences that you find in the sequence, but it's also the direction in which you're reading it. So because you have a 5' end and a 3' end, you have direction. And this direction gives you information in a very specific way. So you have TGCA on the left side of this double helix. But if you read it again on the right side from 5' to 3', the sequence you find is T, G, C, A. So because we have a molecule that is both anti-parallel, meaning the both sides are opposite, and you have this matching business going on where the T matches with an A and a G matches with a C. The left strand and the right strand, no matter how you read it, whether you read the right strand, the left strand, or the right strand, you get the same sequence because you have to start reading from the 5 prime. So the 5 prime reads T, G, C, A, and the right strand reads T, G, C, A, giving you exactly the same sequence whether you read the left strand or the right strand. Not because they're the same, but because they're opposite and inverted. So because this one's upside down, notice the orientation's upside down, you're reading the same sequence no matter which way you look at it. There's some more important things that we'll learn about later as we get into replication and protein synthesis that has to do with this orientation, this direction of the DNA molecule, this 5 prime to 3 prime. As we start talking about replication, as we start talking about DNA transcription or RNA transcription and RNA translation, or protein translation, you start to really think about how important this orientation is, this 5 prime to 3 prime orientation. And you'll start to learn more and more about it as you move deeper and deeper into the understanding of how a cell replicates and how it functions.